Hey guys, this is Dr. Mobeen. We're talking about the central nervous system. The lecture today is about the cells of the nervous system. We have already talked about the neuronal cells that are present in the spinal nerve and the peripheral nerve, and we have seen their classification based on the type of information they carry and the diameter they have and the myelination they have. So that is a different lecture. This lecture is about the cells that are present inside the brain and the spinal cord. So let's look at them. First of all, their basic classification: there are functional cells that are neurons, and then there are parenchymal cells or the connective tissue cells, which are further divided into multiple types. The most important in there are the glial cells. Why they are important is that they make not only the most abundant cells of the brain, they also make the 50% of the primary brain tumors. Glial cells are further divided into microglia, which are the cells that are immunologic in their function. So some people say that they originate from monocyte. They're still figuring it out. Possibly they originate from mesoderm or mesenchyme as compared to the ectoderm, which gives rise to the primary brain tissue. However, just remember this much, that microglia are the defense cell within the brain. That is very important from STEP's point of view. Then are the macroglia. Macroglia are further than divided, and the most important cells in there are astrocytes and then oligodendroglia. We'll talk about them in a second. Then are the ependymal cells. Ependymal cells, if you remember the embryology of the nervous system, ependymal cells are actually the original neuroepithelium. These are the original cells which migrated, which actually formed in the hypoblast or ectoderm when the notochord did the induction of the neural plate. That neural plate cells were actually the neuroepithelial cells, which then separated out in the form of neural tube. Those neuroepithelial cells, once they have given rise to the rest of the brain tissue, then they finally stop dividing further and they become ependymal cells. Ependymal cells would be lining the brain cavity. We'll talk about that. Tanycytes are the cells that are from the choroidal plexus of the blood-brain barrier. They start from there and they go and they finish at the hypothalamus, and they are related to bring leptin from one place to the other. And finally are the Schwann cells. Schwann cells are the myelinating cells, just like oligodendria are the myelination cells. So let's talk about all of them in a second. Functionally, then we have the neurons, which are present within the brain. So the way to look at the neurons inside the brain are: number one, there is neuron or the gray matter present in the cerebral cortex or present in the cerebellar cortex and so on. So where there is a particular layer, an outer layer of the cortex or the gray matter, and then the white matter, which are the fibers coming in or going out. These are called, that is called gray matter itself, because when you cut a fresh slice of the brain, that area appears gray. By the way, brain has three colors when in a fresh cut: the gray matter, the white matter, and the black or the substantia nigra. So three. So the gray matter. Then are the nuclei. Nuclei are also the cell bodies, gray matter, which is cell bodies, but they are present engulfed in the white matter. So they are bundles of the cell bodies present within the white matter are called nuclei. If the bundles of the cell bodies are present outside of the central nervous system, that is brain and the spinal cord, then these are called ganglia. So functionally, neuronal cells can be forming the gray matter or they can be forming the nuclei. If they are present outside of the central nervous system, then they are called, they are part of the peripheral nervous system, then they would be called ganglia. Now neurons are of multiple types. So most of the books would tell you that neurons are pseudo-unipolar, bipolar, and multipolar. I should put pseudo here. I have added one more, and that is apolar. So what happens is, that is the embryological type. The neuroblast, when they arise, they are originally apolar, then they become bipolar for a short time, then they become apolar, and then multipolar. So apolar cells in the beginning, in the embryological state of the central nervous system development, are present, which are neuroblasts. Now pseudo-unipolar, I believe you have seen the pseudo-unipolar cells. These are the sensory cells that come into the spinal cord, the sensory nerve. Why are they called pseudo-unipolar? Because when you see them under the microscope, they look like they have one pole. However, they are actually bipolar, but their two poles have come together enough that they look like one pole. So ideally, this situation is this. So this is the bipolar neuron, but the two poles have come and merged with each other. So under the microscope, they look like unipolar. So that is why they are pseudo false bipolar. They are the sensory afferent neurons which are present in the spinal cord. So that is the pseudo unipolar. Apolar, embryological. Bipolar, vestibulocochlear cells or the hearing cells, these are bipolar. So they actually are true bipolar. So they have one axon, they have two primary fibers attached with them. So these are bipolar cells. And then multipolar, all the rest of the cells, the majority of the cells in the nervous system and the spinal cord are multipolar. So how are they structured? They have a soma, the cell body. Cell body then has a lots of dendrites. Dendrites are smaller fibers. Most of the time, dendrites are the ones that bring impulses towards the soma, dendrites. And then there is one long fiber which is called the axon. Axon has an initial part which is called axon hillock. It is slightly constricted part. Then the rest of the axon may or may not be myelinated. Now if it is myelinated, then the cell that is going to be myelinating it is either going to be, so this is how the myelination is. Originally this is one cell wrapping it or multiple cells wrapping it. So this is, if the neuron is inside the brain and the spinal cord and does not leave it, central nervous system, then oligodendroglia, these cells are the one that are myelinating cells. Myelination for CNS. Myelination for PNS, peripheral nerves, is the Schwann cells. So this one, Schwann cell, is the myelination for peripheral nervous system or PNS. So oligodendroglia or Schwann cell, SCHWANN, Schwann cells, depending upon PNS or CNS location. Now one oligodendroglia can myelinate up to 30 neurons. So one cell sits in the middle. So let's say this is the cell. It has its branches going out, and these branches can myelinate up to 30 neurons. So this is one oligodendroglia. Okay, so that is myelinated. Here is the axon. Axon hillock is the initial constricted part. Then at the end we have the end button or the terminal, which will mostly, axon would finish on a dendrite. So here is a dendrite, right? So dendrite, and this becomes a synaptic junction. You know this one, that synaptic junction is somewhere where electrical impulse will be transduced to a chemical impulse, which would then again be transduced to an electrical impulse. What that means in simple words is, electrical activity would come here, become chemical activity, some chemical substances are released, which will then again become electrical activity. Now this is not necessary that all neurons synapse on another neuron or dendrite. This could be synapting on the muscle and motor end plate as well. At the end of the day, the function is still the same. Now synaptic junction is present, nerve end is present. The interesting thing, USMLE is really interested in asking you what happens, how things move this way and how things move this way. So that is the anterograde movement and retrograde movement. Similarly, they are also interested in seeing that when the nerve cell is cut, there is a Wallerian degeneration, and what does that mean? So we'll talk about it in a second. Now coming back here, glial cells are microglia and macroglia. In the macroglia we have astrocytes and the oligodendrocytes. So let's start talking about them one by one. Microglia are simply the cells which are the immune cells within the brain. They sit in the brain. Normally the immune cells from outside cannot come in because of the blood-brain barrier. However, when the meninges are inflamed, it is possible that these cells can come in and start acting. But most of the time the injuries within the brain are handled by the microglia. They are kind of monocytes and they do the defense mechanism. Then are the macroglia, which are astrocytes. Astrocytes are mostly the supporting or the connective tissue cells. Astrogliosis is the formation of the scar for the injured brain tissue as well. This is the USMLE question. What cells would give rise to the scar inside the brain tissue? Astrocytes. Oligodendroglia, we talked about it. These are the myelin cells of the central nervous system. Their equivalent are Schwann cells, which are the myelin cells of the peripheral nervous system. Now remember, this schwannoma or acoustic neuroma is a tumor of the Schwann cell which is present at the junction of the cerebellum and medulla. So you say, well, that is the central nervous system area. Yes, it is central nervous system area, but the vestibulocochlear nerve, the 8th nerve, is coming out. It's a peripheral nerve now, and it is covered by Schwann cells, and that is where, when the Schwann cell has a tumor there, it is called schwannoma or acoustic neuroma. Ependymal cells, as we talked before, these are neuroepithelial cells which, after doing their function, giving rise to the brain, becomes the lining of the central nervous system cavities, and they are then called ependymal cells. These cells also are going to be forming this CSF secretion layer, and what happens is that we've gotten the blood-brain barrier. From there we have the CSF or the nutrients coming in through the choroid plexus, and then through the ependymal cell they are secreted into the CSF. Tanycytes, some cells that are present and are going to be bringing the leptin-like transmitters from the choroidal plexus and others to the hypothalamus. Schwann cells, myelination for the peripheral nervous system. Schwannoma or acoustic neuroma is very important clinically for us to know. Now a few things here that you should know from the steps point of view. Number one, the anterograde movement. Anterograde movement is two types. Anterograde movement of substances is of two types, fast and slow. Fast is about 200 to 400 millimeters per day. So imagine that 1 grain of salt is 1 millimeter. So 200 to 400 grains of salt. So if you put 200 to 400 grains of salts, that is how much travel can occur in the anterograde way, and that is called the fast anterograde movement. Normally it is done on the microtubules. All movements are done on the microtubules. The protein is kinesin. This is important. Do not forget. Kinesin is the protein that helps with the fast anterograde movement on the microtubules. Then is the slow. This is fast. There is slow anterograde movement as well. Most of that is about 4 to 5 millimeter. Do you know what is the slow anterograde movement does? This is again a steps question. See, the things that are moving from the cell body towards the nerve terminal are two types. One are the nutrients which are going on the cytoskeleton, for example microtubule and kinesin, and the other one are the building blocks themselves.